Okay, so I'm going to get started for today. So where are we? We're on our website, rockefelleruniversity.github.io. All our videos should be here. I think I'm just missing last week's Friday. Most of the attack. Okay. Maybe not necessarily in the right order. Yes, we have our first attack. I got to put the second attack in. Okay, where was I just now? Let's go backwards here. We go back. Okay, so we have our videos. We've covered some stuff on genomics data, and then we went through how we can handle this genomics data. And then we looked at RNA-seq, how we can process that from FASTQ to aligned differential expression. We did Chipseq, attack-seq. So we looked at trying to identify open regions where we may have activity, transcription factors, polymerases active, bound to the DNA. Now we're going to look at the final major high-throughput sequencing type for bulk data. So that's Chipseq. After this, we're going to look at CRISPR and then some scRNA data as well. So we're going to go single cell soon, but the last bulk really we're looking at now is Chip-seq. So everything we need to run along, we can download the material here, and everything you need to install. It's been pointed out to me, Herpa might have some troubles installing it. I think this is almost resolved. We're just trying to fix something, or we're trying to change the maintainer names, which has caused a problem. But reach out to me directly if you're having any problems with Herpa. You should be able to at least install it from GitHub today, and hopefully Bioconductor Manager install tomorrow. Okay, so then we're into our course. So Chipseq. So actually, Chipseq is one of the things I have done most in my career. Obviously I started in my PhD, I was doing microarrays. Chipseq was when I got my first job. I arrived, and actually one of the first types of high throughput sequencing they were using for this was Chipseq. So you can go back to the very beginning of Illumina sequencing and you'll find some terrible data sets. And these are the original Chipseq. Fundamental in our understanding of, really, I think most of it was to do with development at the time. A lot of it on cancer, epigenetics, but it was one of the first things which was first uses of Illumina sequencing. So it has a lot of depth of experience from many people and lots of tools. That said, Chipseq is probably one of the messier techniques of all the high throughput sequencing technologies. And we're going to talk about it in a slightly quantitative way, but often Chipseq is just used qualitatively. I have a transcription factor binding site here, but we're going to attempt to also use it to say we have more of this transcription factor binding in this condition at this site versus this. So let's talk about what Chipseq is. So Chipseq is chromatin immunoprecipitation followed by deep sequencing. So it's a very established method to allow us to identify genome-wide where transcription factors are binding or where we may have epigenetic marks. So we'll think about that again. Effectively, we may have a transcription factor, and we can have them say in orange here, this is our transcription factor of interest, bound to the DNA by its actions. It's found its motif and it has its binding site. Chipseq will typically then fix, cross-link that transcription factor to the DNA, and then we will fractionate that DNA into smaller sections, and only some of those sections of DNA will have linked to them our transcription factor of interest. Okay, so from here we can cross-link, formalin fix often, bind our transcription factors or whatever's on the DNA to the DNA, fractionate it to get shorter. Let me stop share. Sorry, thank you. Share screen. You'll be able to see my slides. Let me try that again. Can people see my slides now? Someone can just put on the chat yes or no. Thank you. Sorry about that. Thank you for jumping in. So let's do that again. So we have our nucleus, we have our DNA, and we have these balls, which are transcription factors or maybe an epigenetic mark. If it's a transcription factor, we will either formalin fix or sometimes UV cross-link, effectively fix our transcription factors to the piece of DNA they are currently bound to. We will then fractionate that. So from all our DNA, we will fractionate it, break it up into smaller strips of double-stranded DNA. And only some of those double-stranded bits of DNA, those little fragments across the genome, will have our transcription factor of interest cross-linked, formalin fixed to it. We can then use an antibody for our transcription factor and actually pull down not just our transcription factor, which binds to the antibody, but the piece of DNA that that transcription factor was bound to. So that way we can use formalin fixing to kind of attach our transcription factor to our piece of DNA and then use antibodies to enrich the pieces of DNA bound to that transcription factor. So following that, we basically end repair our pieces of DNA. We add a certain A tail to this. And this is going to be important later because this gives us direction 5 and 3 for our sequencing. We know the 5' and the 3' of our sequences. We then add the Illumina adapters, and then we can take that, perhaps amplify it through PCR amplification, and then post that. We can then stick that onto the sequencer and sequence this. Okay, so we've gone from our antibody-bound DNA, and we effectively adapt this DNA for Illumina sequencing, and then we attach it to the sequencer. And then we know we get sequences from this fragment where the antibody is bound, probably just from the 5' end. Chipseq's usually single-ended, but often we will get a bit from the 5' end and a bit from the 3'. So that means that from our experiment, we should then pull out something we recognize well, a FASTQ format of the reads containing, sorry. So we should have then, representing our pieces of DNA, we should have sequences from them in FASTQ format. So if we remember FASTQ, we have our ID, our sequence, and our quality corresponding to the parts of the sequence. So we're going to use some real data again for this workshop. We're actually going to use one of my favorite transcription factors, MYC. And we're going to use it from the MEL and the CH12 mouse cell lines. We can get this from ENCODE here directly. And you can also go get the FASTQ directly if you just download this link. When we do the exercises later on, I'm going to ask you to work with the other replicate. So you might want to download that as well. You can get that from here if you want to get ready for the exercises. So now we're going to work through this just like we have before. It's a little bit more tailored towards Chipseq, but a lot of this hopefully we've seen before. You should be reasonably comfortable with this at this point. That said, we're going to step through it anyway. So if we've got raw FASTQ data, in order to read and handle and manipulate FASTQ data, we need to use the short read package. This is the package we use for Illumina short reads. We should have already done this, and we can go back to our Bioconductor FASTQ session. But to get the short read package loaded, we just need library short read, and that will load it and get it available with all its functions to our computer. So we're going to review the raw sequencing data using short read. We're going to do some QC similar to what we did in RNA-seq, but now we're going to actually use the short read package rather than rFASP. We don't need to review all the reads to get an understanding of data quality. Often, I mean, this is the nice thing about rFASP, it will process all your reads if you want to do quality control cleanup. But if you just want to get an assessment of quality, you can actually just sub-sample your reads, save yourself some time and memory, and get an idea about what's happening in your file. Importantly, if we're going to subsample random reads from a FASTQ, we can't just take the first 1,000 reads in our file. And that's because FASTQ is often ordered by their position on the sequencer. So if we take the first 1,000, first million, we're going to take the top left of our sequencer, or the top rows. We're not going to get a reflection of quality across our entire flow cell run, the entire run for our sequencing. So all we're going to do is retrieve random reads across our FASTQ file. So we're going to select roughly from equal intervals in our file. To do that, we can use the FASTQ sampler function from the short read package. We just need to set up the FASTQ sampler function. We give it the FASTQ we want to sample. So in this case, it's still gzipped and it's in our downloads. It's the one I've downloaded from Encode to follow along. And I'm going to sample 1,000,000 reads, n equals 10 to the power of 6. And once I've built my FASTQ sampler object, when I want to retrieve 1,000,000 reads from it, I just go yield on our FASTQ sampler object, and it returns to me a short read queue object. And this is the short read object we can start to manipulate. Importantly, it's taken 1,000,000 reads randomly from that file. If I was to run yield again, it would randomly sample another 1,000,000 reads. If I just kept running yield, it would randomly sample 1,000,000 reads for me. So what we get then is hopefully something we recognize by now, a short read queue object. And if we just type that into the screen, so we've got FASTQ here. If we just type that into the console, press enter, we get our class short read queue. We have 1,000,000 reads, and we only have 36 cycles. This gives you an idea about where we've come from. Now we regularly do 75, 50, 100 base pair reads. In those days, 36 base pairs was probably the absolute standard. And before that, it was 28 base pairs read, because this is really all we could get back in those days. So if we want to, we can then access information from the FASTQ file using our accessor functions we've seen previously. So we can use sread on our short read queue object. That will give us a DNA string set of the sequences, the actual read sequences. So this part here. If we want, we can get the qualities. So those are the quality lines, the Fred quality score, and we can get that as a BS string set object. And we can also get the IDs. So IDs will be important if we want to do some quality control ourselves, because IDs carry the flow cell name, or there, the machine name, and then the position on the flow cell. Okay, so it's a lot of important information in that ID. We look here, we can see our DNA string set object. We have 1,000,000 because we sampled 1,000,000. We have our sequence and we have our width. So some simple things we want to check in terms of our quality metrics for our newly subsampled FASTQ. First thing we really want to check is our scores for our reads. So the overall quality of an entire read. So if a read has failed, it will have low overall quality. High or have higher quality if it looks good. So what we need to do is first extract the qualities, and we can do that with quality FASTQ, and this will give us our DNA string set of qualities. And then we can apply the alphabet score to this qualities object. This, I think it's a DNA string set still. And then we get back a vector and the quality for each read in the order that they were inside our FASTQ short read queue object. Okay, so this looks good. You can see here we have one read which looks really low quality. Reality, to get an idea about where your cutoff would be, you want to plot the entire distribution of your qualities for your reads. Okay, so remembering alphabet score is just summing your read quality, right? If we got reads of different lengths and we want to compare them, we need to think about normalizing to the read length. But in this case, it doesn't matter. They're all the same length. So we can just make a plot of the distribution of quality scores, and then that will tell us, hopefully, where we can take a cutoff. So to do that, we'll do library ggplot, and I will pass in a data frame of all our qualities. So the vector of qualities from here. So that'll be 1,000,000 long. And I'll get my data frame of 1,000,000 rows. And then I'll pass that to ggplot. I'll give it the column I want to summarize for a histogram. And here it's going to be the read queue. It's the only column we have. Tell it to do a geom histogram. And we'll do theme minimal just to give us a nice kind of looking plot. And if we look at this, I mean, immediately we could say we could just take a cutoff somewhere here at the base. So like roughly around 510, maybe 600, you could take a cutoff. And you would include most of the good quality reads, but you would ditch these really low quality reads we see here. And that's where this read would have sat. So that way we can clean up and maybe get rid of some low quality. The other thing we want to check is the occurrence of bases across time, across the sequencing. And we want to look at the general composition of reads in our sequences. We know that there is a bias potentially, it's called a GC bias. And it's just because sequences with Gs and Cs, I believe, are higher or better to be sequenced, more easily sequenced or amplified as well. So we need to potentially keep an eye out for that. And one way we can do that is by trying to get an idea of our overall content of A's, G's, C's, and T's in our data. Oh, sorry, skips 2. I actually skipped my old talk, sorry. So we can do alphabet frequency and alphabet cycle to get some of this information. First thing we're going to check is alphabet frequency. So what we're going to do is extract the sequences from our FASTQ by using sread on our FASTQ object. We get our DNA string set here, and I can just pass that to the alphabet frequency function, and that gives me a matrix now, and this matrix will be the same number of rows as there were reads, right? So we get a value for every read, and then we get the columns of all the potential bases. And we can look along and I can say, well, my first read is rich for T, maybe this read is richer for C. Okay. So we can get an idea of how often we're seeing any bias in particular reads. But when I get an overall idea on this, I can actually do cull sums on this matrix here. And I'm just going to summarize the main columns, A, C, G, and T. If I do that, I can then extract just the vector for them. Or I'm going to include N2. So I'm going to do the cull sums of the whole matrix and just extract by the names in the vector A, C, G, T, and N. We can see we don't have that many N, but actually here we have slightly more A's and T's than we do C's or G's. So a little bit of a bias overall in our data. A really useful thing to do is to see if you have a change or a bias in the usage of particular bases across time. And this might show you a bias. So it might be that you have always a particular pattern of bases at the beginning of your read. This can happen through systems where there's a selection for a particular motif, for, say, the cutting or the fragmentation. You might always see some bases at the beginning. You also want to look at the end of your read, because that's often where you'll have primers overlapping your actual sequence. So when we look at this, here we can see we have indexes and primers. Sometimes when we sequence too much, we sequence through the insert and into the Illumina primers, okay, adapters. That's actually not a problem. When we do alignment, they're going to get short clipped out. Right? They're going to get soft clipped out. They won't be included. But it's often important that we know how much contamination we had before we start being happy with our sequencing data. And we can optimize it for the next one. So we're going to do alphabet by cycle, and we'll give it our sequences. And if we just look at it here, we can see in the first base, maybe A's look a bit more popular. And then that seems to kind of come down a bit. Maybe it stays quite high for a while, though. But certainly a bias for A's and T's across the cycle. We often want to visualize this because we're not going to be able to get a good display here, but we can visualize this just by rearranging our output here into something like a data frame, which can go to ggplot. So what I'm going to do, I'm going to take our object here, and I'm going to select the A row, the C row, the G row, and the T row, and that's going to give me this kind of row of data here across cycles. I'm then going to concatenate that together, and I'm going to put it in a data frame alongside the cycle, the position of the sequencing. And I'll do that by doing 1 to 6, and I'll repeat that 4 times. And I'll do the same kind of with the A, C, G, and T. So we have bases associated to our frequency and cycle position, except here I will do each equals 36. So just to show you the difference here, remind us about this rep function. So if we do rep C 1, 2, 3, 4, okay, so that's the subtle difference there. If I do rep 1, 2, 3, 4, and I just put 4 as the number, it will repeat this whole vector over and over again 4 times. If I put each equals 4, it does each element of the vector 4 times, and then the next element, and then the next element. So putting that together, we then get our data frame, which effectively contains frequency, the cycle, and then the base at that particular cycle. We can then pass that to ggplot here, and we'll give it the data frame we just created. We'll tell the y to be the count, frequency, x to be the cycle, and then we can do color by base. We'll do G online, and again, theme BW here rather than theme minimal. But what you can see straight off is there is a bias here. There is some type of GC bias, because actually A and T are better sequenced than the C's and the G's. And we can also see there's some bias at the beginning a bit here as well, where we have more A and more G than we do the equivalents in T's and C's. So that's something to keep an eye out. And if you saw that in all your samples, you would think that's just an effect of your methodology. If you saw something strange in one of your samples compared to the others, then you might want to investigate why that sample is different. We also want to get the read quality over cycles, and this is going to allow us to identify whether there were issues with quality dropping off over time. Less so now, but it still happens now, to be fair. As you run out of reagents during the sequencing operation, you start to get less confidence in your bases, so sequencing isn't working so well. So getting an idea of the quality over time, over the cycles of your sequencing is important. We can turn our read qualities we extracted all the way earlier on here. So this gives us our qualities as a BS string set of some sort. We can actually turn that straight into a matrix of quality by read and by cycle by using the slightly strange thing, which is as, sorry, these link to our old courses, as read quality, and then just pass it, but we want it to be a matrix. So I pass the read qualities to this as function. I tell it I want to be a matrix, or it should be a matrix. And you get a matrix here now where we have 36 columns representing the cycles, and we just have the first 2 reads here, first 2 rows, showing the quality across. To get an idea of this overall, how my qualities look across my cycles, I can actually pass this just to the box plot base plotting function. Because the box plot function just takes a matrix, summarizes the columns, and puts them into boxes for you. It's a little easier than ggplot. But we can see exactly what we might be worried about. We can see that the qualities, or at least the distributions of qualities, is getting wider as we go along. But perhaps we wouldn't be too worried here, right? We still have reasonably good median quality towards the end. This is expected. if you saw this dropping off very early, maybe again, and then one sample versus the other, you might want to go talk to your genome center and say, does this look right? This is exactly what I was just saying, really. How are you actually going to set your cutoffs for this is tricky. You're going to have to gain experience with quality control to get an idea about this data looks bad, this data looks good. You will always have a colleague or a collaborator who can help you. But the big recommendation here is to run your samples, your quality control side by side, and look for samples which stick out from the others. Once you have a set of samples you believe are good, use them as your positive control to compare your new samples and your new FastQs to. Build up your own framework of quality cutoffs. We may want to actually filter some of this data. So to filter this data, we're not going to be able to use FastQ sampler. Remember, that only takes 1 million random reads from your data. We probably want to use all your data, but we can do this in a reasonably memory efficient way by using this function FastQ streamer. So FastQ streamer, we just need to give it the object we want to work with, tell it how many reads we want to do at a time, and here I'm just going to say 100,000, and that will produce a FastQ streamer object. Okay, so again, this is different. FastQ sampler ran through your whole file, and at the end wrote out the you yield, and you get 1 million random reads. This will read a certain number of reads from your file, stop, then we can pull out the next 100,000 reads and the next 100,000. So this is just going to allow us to read 100,000 at a time rather than the full FastQ into memory. So to do this, we're just going to show you an example here. We'll set some things before we do the loop. So we'll do total reads equals 0, total reads filt equals 0. And then what we'll do is we'll set up our while loop, and we'll do it while the length of our streaming the data from our FastQ streamer. So this is going to pull 100,000 reads. While we can still pull reads from this object, so while the length of the reads we're pulling from this object is greater than 0, carry on doing the loop. So what's going to happen is it's going to loop through, it's going to pull 100,000 until it gets close to the end, and then it will pull out as many reads as it can in that last one, so maybe 50,000, 40, 20,000. Next time it comes through, it will try and pull out some more reads, it won't get any, and then the length will be 0, and this loop will stop. While the loop's going, it's basically taking the total reads so far, which was 0 here at the beginning. It gets the length of the FastQs it's pulled out, so that should be 100,000 until the last cycles. It's going to filter that by a little score here, so it's only going to take reads with an alphabet score better than 300. It's quite low, looking at the distribution we looked at earlier on. It's also going to take out reads who have an alphabet frequency for n greater than less than 10. Oh no, it's only going to keep reads with an alphabet frequency less than 10. So effectively, it can't have too many unknown bases. And that will be our filter 2. And then we will just add that up to build our overall filter. And again, that will be 0 plus the length of this the first way through. And then we'll write our FastQ to this filtered FastQ.guz. Importantly here, and the last important thing on this slide, is that we're going to do mode equals A. Again, mode equals A is append. Right? So that means that we can open the file and we just keep on writing to it. And eventually, once we've written all the reads, this loop will end, and actually R will just close the unused connection. So we'll have a file with our reads filtered by alphabet score and the frequency of ends, while we're also keeping track of the total. So that's how we can clean up our FastQ and get some qualities like we had previously done. Next thing we need to do is align our Chipseq data. So Chipseq data is typically single end, just its nature. Following our assessment of read quality and any filtering, we're going to align this to our genome of interest. We've already seen that we can contiguously align to a reference genome using some base aligners, things like bow tie, the align function and subread. We don't need to use things like star or subjunc or HiSat2, because it's going to align to the genome and not to splice junctions here. So we want to produce a BAM file. This is our BAM file, right? So the first thing we need to do is create our reference genome. We should have done this before. Here we're going to use the BS genome object, mouse musculus UCSC MM10. You can come to this with your own FASTA as well. And I'll show you the point where you could. So to get our genome from the bioconductor, we'll load the library first, library BS genome mouse musculus UCSC MM10. We'll then have the mouse genome available to us here, and we can see various information on it, including, as you remember from our bioconductor lectures, the actual information on how we can access this. And then we just need to create, this is mouse, so the major chromosomes we want to extract our sequences from. So I'm going to take chromosomes 1 to 19, X, Y, and M, and then going to loop through this character vector, extracting the sequence from our object using our double square brackets and the chromosome name here. And then once I've done that, I'm going to assign the chromosome names to our list of DNA strings. And then I'll take our list of DNA strings and make it a DNA string set. And it's this then, which has our full sequence for our major chromosomes for the mouse MM10 UCSC. See that here. We then just want to write this out, because some of these aligners like bowtie 2 can just take the object, but I always want the FASTA written out because I want to be able to share it with people. It can't just be an object stuck inside my computer. So I'm going to write X string set, and I just need to give it my DNA string set object from here, which is named. And then I create a FASTA file. I just need to put the extension .far or .fasta, and it will know to make a FASTA file from this, and it will call that FASTA. Okay. You've got a FASTA file already. You know, this is the point you could use your own FASTA file after this to make your alignment, to make your indexing. So we're going to be using the align function, not the subject function, from the folks behind subread, so from our subread. But before we do any alignment, we need to build an index. Okay, so subjunc and align would actually both use the same index, so that's fine. We just need a FASTA file, so we've just produced one. We produced this one, BS genome musculus UCSC MM10 main.cur.far. So we're going to load the library R subread, and I'm going to use this FASTA to create an index called MM10 main.cur. Importantly, I'm going to set two things here. Index split equals true. So it's going to split the index into smaller chunks for me. And then I'll do memory equals 8,000. So then it's going to use 8 gigabytes worth of memory to build these chunks. That means when I come and do alignment later on, it's also going to limit my memory to a maximum of 8 gigs. Okay. So when we build the index, we're really controlling the memory is going to be used for both indexing and alignment. Like I say here, if you've actually done this already for the RNA-seq lectures, we can just use the same index. It isn't a special index, it's the same function. So it's not a special index for spliced alignment using subjunc versus normal alignment using align function inside our subread. So we can now align our raw sequences to the FASTQ in the new FASTA format. To the genome in the FASTA format. We're going to use the align function, as I said. We're going to do align MM10. I'm going to give it the index. I'll give it our filtered FASTQ. I'm going to tell it the output format is BAM. I actually don't know what the other options are for this. I have to check that. The output file we want to create is micmel1.bam. The type is DNA, so we can specify RNA or DNA there. The number of threads, I'm going to use 4. On your Mac laptops, you probably have 4 threads. So you can use 4 threads for this. Importantly, when I run this the first time, it's going to tell me off. And it's going to say, you've got the thread offset wrong. And that's because it's looking at the qualities within our, it's looking at this quality line. And it's kind of comparing that to an internal map. Let me just see if I can quickly pull this out. There we go. It's comparing it to the internal map of potential scores for FASTQ. So it's basically saying, if I see thread quality scores within this range, then I couldn't be using this purple range of values, right? And that's just because thread quality scores over time, the ranges they include have changed. You can see Sanger had one, Solexa had one, Illumina. Then Illumina went for a slightly different one, and then Illumina went back to thread plus 33. So most of you, as you can see here, the modern Illumina and PacBio all use thread plus 33. It's their standard quality scores. Most of the time, we don't need to worry about this. Some aligners will ask you for the thread quality scores. Subread is one of them. But if you get it wrong, it's going to tell you off and actually tells you what it should be putting there. So in this case, we'll put thread offset equals 64. After it's told me that was the issue, and then we can get on with the alignment. So that's sub-read alignment. The other one which most people or a lot of people would use is R bowtie 2 to use the bowtie 2 package. Quasar, which we come across, does have bowtie in it. The only time I've seen bowtie still used is in some clip analysis, some RNA regulatory binding analysis, because it has a little bit more options. We can't use the Quasar package because it's way too memory hungry. So we can use the R bowtie package, and we can just load library R bowtie 2, and we're ready to go. And with our subread, R bowtie 2 requires the index to align to. Although we can use the subread index for both spliced and non-spliced alignment, it's specific to our subread. So bowtie 2, we need to create a bowtie 2 index for. We can give it the same reference faster though. So we do bowtie build, give it the same reference faster, tell it the name of the index to build, and that will go off and create a bowtie 2 index for us to align against. We can then actually use bowtie for our alignment. Bowtie 2 is very popular for ChIP-seq because it's a good standardized genomic aligner. The annoying things about bowtie 2, which work well on the command line but don't work well in R, is that it doesn't work with zipped files and it doesn't like SAM files. So we need to unzip our files before we pass it if they're zipped. So we're going to do R utils, and that contains a gunzip, so it can unzip our fastq.guz file to just a standard fastq. And I'm going to say remove equals false because I'm paranoid. I don't like things to be deleted once I've unzipped them. I want the original file to still be around. And once I've got that unzipped, I can use the bowtie 2 function, give it our index, tell it I want the SAM output to be called this.sam. And this is my sequence I'm going to use for alignment, the unzipped version of this. bowtie 2 is going to output a SAM file. Luckily, we can use our SAM tools as BAM. And that's very memory efficient. It's going to make a BAM file from our SAM file. And I think it's actually going to index it for us, sort it and index it, but we'll do that ourselves just to be sure. I've said this in previous lectures, but bowtie 2 and lots of these programs, you may have things hanging around afterwards. So here we're going to produce a SAM file, which will then convert to a BAM file. We don't need the SAM file anymore. It takes up a lot of space. It's going to be 10 times as big as your BAM file. In reality, it's probably 40 gigs now. So we can just unlink this file, which is the same as deleting it. And we can do unlink, and we'll give it the SAM file. We'll just delete that and move that. Similarly, we will want to go and unlink the unzipped FASTQ file, because that will also be massive. Like it says here, on the command line, you can stream inputs to our bowtie 2. And that just means you're basically unzipping it while you're passing it to bowtie 2. It's part of the same process. We can't do that in R, so it's a little strange. We have to do it up front and then delete it afterwards. Any alignment, any time we want to make a BAM file, we want to check to see if it's sorted. And if it's not, we want to sort it ourselves. I happen to know that our subread doesn't sort your files by default. It's going to put them out in read name order. So the same order you put them in, it's going to come out at. That can be very useful in some cases. But in this case, for downstream, I want to sort it. So I'm going to do library rsantools. I will take our BAM file from subread, and I will sort this. And I'm going to produce this file. Importantly, I'm not going to put a .bam at the end of it because this is a prefix. So it's going to create a file sr_mic.mel_rep1.bam, even though I'm not adding it. Once I've done that, I got to index my BAM file because the index will create the .bam.bai file, the binary index, and that allows me to do random access, but also I can do index stats and quickly get mapping reads. Perfect. So we've gone and we've aligned our FASTQ to BAM file either through subread or bowtie. Now try our next part, which is to try and get some stats and some very simple. I think we're going to try and make a bigwig. The first thing we're going to do is IDX stats BAM. We use this in our ChIP-seq lectures if you're here, to try and get an overview of where our reads landed. If we do IDX stats BAM on our BAM file here, we can then take out from the mapped column from the result and get the sum to get the total mapped reads. And then I'm just going to do a ggplot of the data frame which came back from this, and I will do X will be our X, so we're going to flip it. Y will be the mapping rate, but we're going to flip it, so this is the Y, and the X will be the chromosome. We're going to do stat equals identity because I already have the total number of mapped reads. We're not building the stat ourselves. And I'll do coordinate. So I think let's just pull up the ChIP-seq quickly. Because in there we have this very different kind of plot for this. This one. So same plot, very different looking result. There it is. So if we look at the signal for a ChIP-seq, we can see we have a big problem there, right? Chromosome M was sucking up all the reads, and actually we know that got better in time. But yeah, that's a problem with the ChIP-seq. You get a lot of mitochondrial reads. ChIP-seq, we don't have that problem. But if you look at this, there is something strange here. Chromosomes go in the order of their size, and chromosome 2 has a lot more reads than chromosome 1. And then 9, 11 seem to stick out a bit. Maybe this one too. Yeah. I can tell you up front, this is because of blacklists. Chromosome 2 has a massive blacklist region. It sucks up enough reads that it makes the whole chromosome look like it's got more reads. Chromosome 9 and 11 also famously have some very big blacklists in maps. We're going to cover that in more detail in our next session, but already we can see the evidence of it just in the fact that we have so many more mapped reads to chromosome 2 than 1, and 11 and 9 versus 10 and 8. So already we're seeing potentially an issue. And if you knew this up front, you know, the extent of chromosome 2 versus chromosome 1 difference, without having to look at anything else, is going to tell you here how much blacklist, how much background signal do I have. Probably one of the things we want to do in these first steps is to get, and especially with ChIP-seq, more so than a TAG-seq and RNA-seq, ChIP-seq you want a bigwig to look at. Because ChIP-seq, as you'll see in our next thing, you have such different range and efficiency, and it's kind of expected. CTCF is 90% efficient to ChIP. MIC is 5% efficient to ChIP, right? So they're going to get very different looking bigwigs. So you want to go against them. So to make a bigwig, it's pretty straightforward. We just got to do coverage, parse it, because we've used this coverage function a few times. We've used it on G ranges, we've used it on GA alignments, and we've used it on BAM files and BAM file objects. Here we're just giving it the path to the BAM file. We go coverage, and this will give us these RLE list objects we've seen before, like this. We have the coverage across the whole chromosome compressed into this runs. We have 33,000 run of 0, and then 50 runs of 20, and then more 0. This was what we will get back from our coverage here. We'll get our RLE list object. And then if I want to export that, I can just load the R track layer package and then do export.bw bigwig, give it our RLE list we just generated here, and tell it the connection I want to write to .bigwig. We know we can do a little bit better because we could probably take into account the number of total map reads, and we extracted that from our IDX stats earlier on. If you're going to ever do this normalization, always get it from IDX stats because it's the fastest possible way. And therefore, I will just now give it my BAM file, but I will give it additionally a weight, and I will do 1,000,000 reads divided by the total number of map reads. So the more map reads, we should be down weighting our BAM. This will give me our RLE list object, and now I can export the normalized RLE list object to our connection. It would be our weighted bigwig. Okay, so then you'll get this final, this is our BAM and our bigwig from Mick. But just to point out something which is going to be important in our next session, if you look carefully, so this is our positive reads, our reads on the positive strand and the reads on the negative strand. If you look very carefully, once you get a composite, it looks okay. It doesn't look super smooth, but it looks okay. But if you look, you can actually see the positive reads are to the left of the negative reads, right? You see that? I think so. Right. And it's that feature of ChIP-seq which actually is one of the biggest things in quality control. You want your reads to be piled up, positive on the left, negative on the right. We're going to look at that in our next session. We even got a little animation to show us what we can do with that. Okay, so that's the end of the session one. Have some exercises here for quality. Feel free to run through them or not. I'm going to give you the answers here as well. You actually got the R code as well. Okay, let's go all the solutions so you can see a full solution for an alignment and then exporting of a bigwig. And then what we'll do is we'll come back into our next session after the break, and I'll run through some very particular ChIP-seq quality. We'll talk in detail about the blacklists. We'll talk about the shifting of positive over negative. I think that's about it. So we'll start that at 3 o'clock. If you want to go somewhere and do this on Zoom, feel free. I'm very happy with that. If you want to hang around, I'm also super happy. Geez, let me try that again. Okay, thank you, Kelsey. Let me try that again. Okay, so we're going to come through our session today. I'm going to repeat the first 4 minutes. So we're going to be using the MIC Chipseq. We've already looked a little bit at this. We, I think, used MIC-MEL replicate 1. MIC-MEL replicate 2 was for the exercise, but we're going to need a few replicates and a few different groups if we're going to do our differential Chipseq later on. So we're going to take MIC across 2 cell lines. Because it's encode, they always have 2 replicates, so we can do some type of differential. For quality control, Chipseq has lots of potential sources of noise. Sorry for the people in the room, you, sir, who've already heard this. And that includes things like varying antibody efficiency. CTCF is 80% efficient, around that. Cohesin is 50% efficient. Most transcription factor Chipseq is actually probably 5% efficient. And that means 5% of your signal lands in regions we're going to term DNA transcription factor binding. Okay. That's a hell of a range, 80% down to 5%, and even lower, 3%. You might still believe that to be acceptable chip. Non-specific binding, DNA is sticky, and your antibody is sticky. You may pull down non-specific regions or regions not related to your actual transcription factor. It's just background. We have problems with library complexity, and that includes really coming in with too little sample, not enough sequence, DNA sequences to actually sequence. That means you end up sequencing the same sequence over and over again, and you end up with sequence duplicates. A few regions of your genome being very well sequenced, but actually it doesn't represent the DNA which was bound by your transcription factor. So we can detect that through duplicates. And then we have this idea of chip artifacts, background, and really, where's my camera's off? Really, that relates to the blacklists. And these are regions of the genome which just suck up signal from chip and attack, but not really related to the source of the Chipseq. So you do cohesin or CTCF or polymerase, you still see signal in these particular regions. These are our blacklists. So we're going to use we're going to discuss a few of these quality metrics now. For more review yourself, you can go look at the ENCODE quality metrics, Georgi Marinov, Anshul Kandarji, Peter Park, Barbara Wold, or a really good paper by Barbara Wold again, and Shirley Liu, who wrote MAX, the peak caller we use. This looks at overestimation of artifact duplicates. So I just said duplicates are related to library complexity. Actually, nowadays, if you sequence too much, you're going to end up with duplicates anyway. And these people have looked at this and basically saying we overestimate it. That's going to be useful for when we start to do differential binding, and we're going to talk about that later. The final paper is actually by myself, and that was just when and what QC is useful. And really, we did this just so that we could have a reference for our package ChIP-QC. But it does talk us through why we remove certain portions of our data and when we should do these QC. Before we get any further, one thing which differs our Chipseq from most other types of analysis we're looking at is we always want something like an input control, an IP control, we call them. So these are typically input samples, so the same sample that would have gone through fragmentation and then antibody capture. You just leave out the antibody capture step and you have a sample which really represents your background. Okay, so when you sequence that, that should be your genomic background. And then when we have our actual sample, which is IP'd, we should see enrichment in regions over the control. And those are our transcription factor binding sites, where we have more DNA because we've actually enriched for antibody. This is going to allow us in some way to control for artifact regions, because there are regions which are going to have signal no matter what we chip, or even if we're an input. So we can allow, we can actually detect these regions in our controls. Never run a Chipseq without considering what input to use. Okay, and it can be complex, but an example would be if you're doing tumor samples for Chipseq, you want a matched input, right? Tumor samples are going through all sorts of DNA rearrangements. This may cause problems. You do your DNA in your actual sample isn't quite the same as the genome we usually align to. Having a matched input is really important for that. If similar differing tissues may share a common input, right? If you have different conditions. Here in this case, differing conditions of the same tissue, you may want to share a common input. Okay. Most important thing is you sit down and you try and decide what is my actual background for my experiment. Usually it's matched to either your tissue type or your cell type in terms of things like cancer. So the ChIP-seq package, which is what we wrote and that paper's about, it really wraps some of the metrics which have been circling around at the time into a bioconductor package. What was important is we need to think about when we take these metrics. If we clean up our data first and then take these metrics, obviously data looks great. So we need to go in with a reasonably unfiltered BAM, uncleaned up BAM file. And we can use this function within inside the ChIP-seq package, ChIP-seq sample, to try and run some generic QC, ChIP-seq on our data. So this is going to take a black list, and a black list is a bed file of G ranges and genome names, genome name. But effectively, it's just a bed file of sites which we believe for a species will have a lot of signal. So I'm going to open up the page here. Here we have the one for the blacklist for ENCODE. If you go to file details, you'll see we can download it for various genomes, only HG19 and MM10 at the moment here. You can download them as big bed or bed, and then that's what we can import as a G ranges and use for our filtering. I think there's actually, if you go to Anshul Kundaji's site, maybe it's still not the same page. Yes, so he has new updated human ones, but he also has ones for some other species as well, worm, fly, which may be useful. If you're struggling to find your own genome here, let's try Grey List. Am I going to pull up the new Netflix movie? Grey List, Chipseq. Grey List chip. We also have, this is from old, well, it's the same person who wrote ChIP-seq with me, Rory Stark, but it was actually written by Gordon Brown. And that effectively allows you to create your own artifact list for your own genome, your own data, just given control input samples. Well, we've got a blacklist. We're working on MM10, so we can pass it the blacklist I just downloaded. I have to tell it the genome's MM10. It's going to look up some annotation from the Bioconductor databases. Let me just give it our BAM file and pass that to the ChIP-seq sample. So here we actually have it. We actually have a working example using the data we just downloaded. So we download that blacklist from MM10 I just shown you. I'm going to give it the sample we aligned earlier on in our first session. So this is the one from subread, and it's sorted, and it will have an index. We need a sorted indexed file for ChIP-seq. And we can tell it if we don't want to run it across all the chromosomes, we're giving it the blacklist there, but if we want to run it just across a subset of chromosomes, we're just going to run chromosomes 1 to 10, and I can set that there. And it's just going to save us a bit of time. I recommend running it across all your chromosomes, but if you're in class, just run it across a few. And if we look, we have then a ChIP-seq sample object from the ChIP-seq package. So if we display this, if we just type in ChIP-seq into the ChIP-seq object into the console, it will display something reasonably useful. It tells you the total number of mapped reads across the chromosomes we're looking at, the number of reads which pass MapQ filter. MapQ is our mapping quality, so quite a lot of considered multi-mapping here. Percent of reads which land in our blacklisted region is around 8%, so that's quite a lot. And then we have various other metrics we can look into, including things like the proportion of counts across various regions. So it's just off the side of the page, but it would be regions like TSSs, promoters, exons. It's always best, like we were talking about with looking at your FASTQ quality control, it's always best to run your samples together. And then you can compare how good do my metrics look in one sample versus another. Does it really stick out? So we can do that a few different ways in ChIP-QC, but I think this is the way I prefer. So here we're going to run our BAM files here, which we've aligned and sorted and indexed. And I'm just going to apply that. Here I'm using BPL apply, so that's a parallel version of apply. You can just use lapply. Give it the BAMs, tell it I want to use the ChIP-QC sample function, and then I can give it the various input functions for ChIP-QC sample after the comma here. So I'm going to give annotation MM10. The blacklist we just set on the previous page here. And then again, I'm going to tell it the chromosomes to do it from. And that gives me a list of ChIP-QC sample objects, and then I just assign names to them. I'm going to assign the name of the BAM file. So now I can do something really useful. And now I've created a list and named them. I can pass that to a new function, QC metrics, and it's going to give me a table of all the relevant metrics. If we look here, you can see we have the number of reads, the filters. So that's actually to do with multi-mapping, the MapQ filter. The read length, you can see that one of them is actually 36, which is slightly strange, but it's the input for this sample. We can look at the fragment length. So even though these are single end, we use some tricks to predict fragment length. I will show you in a few slides. And we have our Ribble here, which is our reads and blacklists. And you can see it's quite high. It's like above 10% reads and blacklist. So that's a lot of your data just going somewhere we're going to throw away. One of the first things we want to do is try and predict our fragment length. So we only have single end read. In AttackSeq, as we saw, we can actually get our fragment length by looking at where our read 1 and read 2 aligned. The distance between them tells them our fragment length. Here we're going to have to predict it. And fragment length prediction is really an essential part of peak calling. It's also an essential part of summit identification, which is useful for motif calling in ChIP-seq. And we need it for our coverage profiles. If we're going to make a bigwig, it's better to reconstruct the full length fragment for that bigwig rather than just plus our reads. So there are kind of two ways to predict fragment length. Well, there's probably more than two ways, but two major ways are cross-correlation and cross-coverage, which effectively allow us to assess how much the reads are clustering by strand separated from each other. This isn't just going to be a way of predicting fragment length, but we can also use this method of cross-correlation, cross-coverage of the reads on the different strands to give us a really good metric of quality. It's one of the major metrics of quality from the ENCODE consortium, and it's very useful quality report. The quality metric we report in ChIP-QC. Okay, so just to talk us through this a little bit, given again our DNA pieces which have in triangle here would be our transcription factor of interest. We use our antibody and we can pull out just the DNAs, in an ideal world, just the DNA which was bound to our transcription factor by using antibodies. And we go from 3 pieces of DNA here to 2, and those 2 pieces of DNA would have been bound by our transcription factor. Through a process of then end repair, hopefully you remember A-tailing and adaptive ligation, we get to the point where we have something ready for PCR amplification, and then after PCR amplification, we can get ready for sequencing. So the important part here was this A-tailing, because if you look here, you can see the A goes on the ends of the different strands differentially. Right? It goes on the end of the top strand here, and then it goes to really the front of the bottom strand here. So we can actually tell if we're sequencing the top or the bottom strand, the negative or the reverse strand, positive and negative strand. So ChIP-seq typically is short single end reads, and because I think it was one of the first sequencing we did, kind of stayed short. It was around 36 to 50 is about the longest you'll get from ChIP-seq. Because it's double strand and single end sequencing, we can actually tell the 5' end of the fragment, so the 5' end of the DNA will be on the positive strand, and the 3' end of the fragment should be on the negative strand. Okay, so we should be sequencing this end on the positive, and then the negative should be here. Although we only have a partial sequence of strand, with predicted fragment length, if we knew how long the fragment was, we could extend our read to the fragment length, and then we would have the whole. To do this, we would extend reads only in the positive direction for when they're positive, and reads only in the negative direction when they're negative. So we take a positive read and extend it to the fragment length that way, and negative read on the other strand, we extend it. So if we look at this, this is just kind of an idea of the fragments we have pulled down around the transcription factor binding site. So let's say this blue is the transcription factor binding site. And if we sequence the ends of the fragments coming down from this, each one of these could have been bound. So each one of these fragments could have been bound by our transcription factor. If we were sequence just the ends, which is what we do in sequencing, all the positive reads coming from the 5', the left end, would end up on the left-hand side of our transcription factor binding site. If we sequence the other side, and they will all be negative, we will have all our negative reads on the other side of the transcription factor binding site. So actually, just looking at this now, the mean distance between the red parts here and the green parts, the positive reads and the negative reads around this transcription factor binding site actually gives us our fragment. By taking the mean of where our reads aligned here on the positive and the mean of the reads aligning on the negative, we get an idea. The transcription factor binding site must be in the middle, and the distance between them is probably our fragment. So the reason that this is useful, you can see here a really nice demo. So this is genuine CTCF data I took for our course. If we just plot the data as positive and negative reads with real data, and we look at the motif, we don't know where the transcription factor actually bound. We know where the motif is for CTCF, so it should be bound there. We can see that, yes, the major bit of the positive signals on the left, the major bit of the negative signals on the right. And then if we were to extend them to the real predictor, to the predicted fragment length, you can see that actually they fall on top of each other beautifully. And that looks like a real peak, right? Rather than a mess of signal, an uneven left-handed mess of signal for Watson strand, right-handed mess of signal for the Crick strand. So how do we actually go about using this kind of idea? The positive reads will be on the left of the fragments, negative reads will be on the right of the fragments, and fragments will pile up around transcription sites or over transcription sites. We can actually do this concept of correlation analysis. I'm going to restart this slide, I guess. We'll have to wait for it to kind of go through. Never quite sure how to restart the slide. So along in this graph here, what we have is on the top in the red, we have the positive reads, and on the bottom we have the negative reads, just for this tiny locus that we're looking at. And what I'm doing here, we'll probably just wait for it to get to the end of it. What I'm going to do here is take the positive reads and shift them one base pair at a time to the right-hand side along the genome. And every time I shift the reads, I'm going to look at the correlation between signal on the positive and the negative strand. So right now, it's obviously very little correlation. So if we start before the reads have shifted over, we can see the correlation is low. And now we can start to see that the reads are kind of coming over each other. Correlation starting to get bigger. And now they look almost perfectly lined up, and we're probably going to hit our maximum point of correlation. And then once we shift the reads past the positive reads past the negative reads, we can see the correlation starting to drop off. Okay. So the point of maximum correlation there is our fragment size. That is the distance between our positive and our negative reads. And although I'm just showing you for one peak here, what we actually do is shift this genome-wide. We take our positive reads and we shift all the reads on our positive strand, one base pair. We take the correlation with the negative strand. We do another base pair and we take the correlation with the negative strand. And we do that for a range of base pairs, probably 400 base pairs, just to get all the potential fragment lengths we really expect to see. And then we look for the point of maximum correlation, and that's our fragment. So this is correlation. You can also use coverage. So you could say coverage is slightly different. We're really looking there at the minimum coverage. So once these things overlap the most, the actual coverage of the genome is reduced because you're overlapping. So correlational coverage, you can use the maximum or the minimum to define the end. So that actually is, although that's a way of predicting fragment length, it's also a way of giving us overall quality. Because if your data is arranged as peaks, where your left-hand side is like your positives are on the left and your negatives are on the right, we expect there to be a point of reasonable correlation when we overlap these. Okay? If your data wasn't arranged like this, then we would never see an increase in correlation and it would just stay flat. So we can actually use the degree of correlation at the maximum, so at our predicted fragment length, versus the minimum correlation in our window to give us what is called the fragment strand cross correlation. And that's our first really useful metric. Okay, so the maximum correlation versus the minimum correlation in the window we shift over gives us an idea of score. Okay, if your peaks are arranged like this and mostly like this, your correlation is going to be very high. If your peaks are just background noise, your correlation will be good. We also have something kind of interesting here, which is this first peak. Now, this first peak is a bit of a mystery at the moment, right? But where's that come from? Well, that first peak is exactly your read length. And it's termed by the ENCODE consortium your phantom peak. And we get a phantom strand cross correlation there as well. So whereas this fragment strand cross correlation, the score between here and your background gives you an idea of your quality, the phantom peak is actually a measure of your artifact. So the distance between this peak and there, your measure of your background. So we can generate these very easily in ChIP-seq. It's already done the calculation when we ran the ChIP-seq sample function. We just need to run plot.cc on our object, our FastQ object. And we're going to tell it here, facet by sample. So we'll get one by every single sample. Already you can kind of see the really good chips. This chip looks like it's really good, CH12-2. These ones look pretty good. It's quite high, the signal, but the phantom isn't much lower than the main peak. And what's nice is if we look at the input, where we hope not to see too much signal around the fragment length, because the input shouldn't have many peaks. It's the input control. We haven't had an IP on it. We can see here that the phantom peak is much higher, or it's higher than this peak here, much higher than this peak here. And that's telling us that this input's probably pretty good. If you see just the phantom peak and no presence of this, or the phantom peak higher than your fragment peak, your samples, your ChIP-seq isn't looking great. We can also include metadata as a data frame where the first column is our sample names to allow us to group our plot in different ways. And this extends through all of ChIP-seq. That's really the benefit of using ggplot. We just need to create a data frame where the first column is our sample names, and then the other columns are metadata we want to include in our plot. And now I can run plot CC, and I can do facet by, in this case now, tissue, because I've added additional metadata here in add metadata, my meta. I can also color by any other columns I had in there, so I'm going to color by antibody. Now we can see tissue looks pretty similar, maybe a little bit more variable in the CH12, while MEL looks pretty tight. Also quite variable in the difference here between the input phantom peak and the main peak compared to MEL. But importantly, we can add other metadata to our ChIP-seq plots. And then because this is a ggplot2, and this is why we want to return ggplot2 objects, we can also customize it to make sure it's the same theme and style as our other plots. So we can just do plus theme BW and then GG title, and we finally have a nice ggplot, which looks a little prettier than perhaps that. I also somehow changed my orientation of this, maybe. Okay, so we talked about blacklists a fair bit. ChIP-seq will often show the presence of these common artifacts, these ultra-high signal regions. And as I said previously, it's Anshul Kundarji who's kind of gone through all the ENCODE data to find these regions by observation. But just to show you it here we have polymerase ChIP, so that's for polymerase 2. Should expect that to see around promoters, and here's our MIC ChIP-seq. And if we look, this is one of the blacklist regions here for MM9. And you can just see the signal looks the same across these regions, irrespective of what I've chipped, right? And it's actually super high as well. So there's a lot of signal here, and it doesn't matter what I chip, I'm going to get a lot of signal there. That is what the blacklists look like. So blacklists, I can tell you from our investigation, affect many of these metrics. And in fact, if we were to wonder what that phantom peak came from, what I did in our paper is I just split the data up across a few thousand samples. And you can measure the cross correlation across blacklists, across duplicates, and across peaks. And just aggregating across ER, we can see that we only see a fragment length correlation for peaks. But when we do the cross correlation across these blacklist regions, we can see it's all phantom peak. I'm not sure if I'm going to go into much more explanation of why, but it's to do with coverage in the genome. There are some places which just can't be mapped or are mapped very repetitively. And that's what leads to this read length peak at the peak at the read length. So we can tell here it's come a lot from the blacklist. We also know that SSD, which is standardized standard deviation, so it's just the standard deviation of signal across your genome. This is also a measure which is really sensitive to blacklists. And it's a measure which has been put forward as a way of assessing your ChIP-seq quality. But if we look here, what happens to our SSD scores, and what I've done is I've taken all the ENCODE data which was available at the time, so a few thousand data sets. And if you look at the distribution of SSD scores before and after removing the blacklist, these are the 3 different blacklists. You can see it's a totally different range of signal. And that's the issue here, is that if you do SSD before blacklisting, all you're detecting is the blacklist. If you do it after you remove the blacklist, you start to get an idea how good your data actually is. So here, if we look at this bottom one, this is after blacklisting, and you can see the inputs are flat. They have very low SSD, which is what we want. Transcription factors are a little higher, and then the histones are also a little higher. Okay. So we have to remove the blacklist before we can use this SSD metric. So that's something we'll do in ChIP-seq for you. We actually kind of remove it internally, and we'll give you the SSD before and after removing. The pre-blacklisting is in red, and you can see a lot more variability there, and the post-blacklisting here in blue. To get this plot, we just need to do plot SSD on our ChIP-seq object. I'm setting some limits of the XLIM here, just so the plot looks a little bit more pretty. If we want to focus in though on our just our post-blacklist scores, we can actually see that we have a really higher blacklist for the input signal. Right? We have a high score here for this input CH12 band. So if we go back, we can also see that the input CH12 band had this massive peak in our cross correlation. Right? So this is the input for CH12. Okay, so that already told us that we had a lot of background signal. And if we believe this plot, that the background signal comes from the blacklist, then it makes sense then that when we look at this post-blacklisting, perhaps we haven't managed to remove all the blacklist. And there may be somewhere in there where we need to do further removal. We might need to run the graylist chip tool and try and identify new artifact regions. So I talked about library complexity, and this is often an over-amplification. It often comes from the over-amplification of reads during the PCR step. And it can lead to a large number of duplicates. And then when you call peaks, you actually call these duplicate regions as if they're a peak. And they're not, they're just low complexity regions. So we want a Chipseq peak to look a bit more like this. And this is kind of like the Chipseq peaks we've been looking at so far, where they are kind of not just a stack of signal. There's a bit of a peak. Right? It's a little bit more building up to the signal. If you get stacks like this, where you just have all your positive reads and negative reads just going straight up on the thing, it's usually caused by over-amplification, just PCR'd the same sequence over and over again. And those aren't the peaks we want. So we can get a nice sense of duplication in our data just by using our flag tag counts function from inside ChIP-seq. The nice thing about flag tag counts is actually going to try, or ChIP-seq is actually going to calculate duplicates from the reads itself. Often you would have to run a separate program to mark duplicates in your data. ChIP-seq will just calculate it from your data itself. We just need to run this flag tag counts function from on our object. And we saw a little bit about quick band flag summary. I think we saw in our SAM tools before. Just looks at the SAM file. And here we're just checking the number of duplicates as called by ChIP-seq divided by the total number of mapped reads. And what we can see straight away is that the inputs have higher duplication rate. Maybe a little bit of an issue, but this sample here, MICMEL1, also has quite a high duplication rate. It sticks out from all the other ones. And then you might want to go back and start to look at it from other metrics to see if there's something else which will stick out about it. Look pretty good here. One of the final things we want to check, and this really depends on your knowledge of your transcription factor, but we might actually want to see where within genes or around genes does our transcription factor signal, our ChIP-seq signal come from. If we're doing a ChIP-seq and we know that this is an antibody for which goes across the gene body, we want to see enrichment in gene body. If it's a transcription factor defined with the promoter, we want to see the promoter. If it's Pol2 serine and it goes all the way to the end of the gene, maybe we want to see enrichment in the UTR regions, 3' UTRs. So we can get this kind of distribution very easily from our ChIP-seq object. We just need to pass our list of ChIP-seq samples to plot_regi, and this again gives us a ggplot, which shows us the relative enrichment for signal across samples. And actually, this looks really nice. We can see we have this sample here, CH12-1, that's slightly lower. CH12-1. I don't remember seeing anything particularly bad about it there. It's hard to tell which one of these ones it was, but probably the less efficient one. But we can see that we have lots of signal at the promoters and the all 5' UTR, which really is the beginning of the gene. So having a lot of signal there is kind of reassuring, and then looking in your inputs and seeing that you have less signal there, that's great, right? We hope to see not much bias for the input around promoters or in the gene, but we certainly see a bias for having signal from our chip samples, our MIC chip samples over that. There is an effect in Chipseq, it's called the SonoSeq effect. And because promoters tend to be more open, as we will know from AttackSeq, that just means that they're also more easily fragmented. Right? So your background isn't truly flat across the genome. You can expect to have a little bit more in promoters, which is why you see a little bit of yellow here. Again, it's called the SonoSeq effect. It's usually not a problem, but worth keeping an eye out. If you saw your input really enriched in your promoters, you need to go look at it in IGV. Maybe you need to run a new input. The next thing we wanted to talk about is peak calling. So we did peak calling kind of briefly before, but we did it with AttackSeq. And we didn't need to worry about things like fragment length because it was paired. So we're going to have to do it a little bit differently here because we have single length. Max2 is still popular. There is a Max3. We haven't used it much. Most people are still using Max2. Max2 itself can predict fragment length, and it does it now in a very similar way than we're doing it. It used to do it slightly differently, and it used to look for just the distance between positive and negative peaks, which were already kind of close to each other. They were trying to give an average distance between what it called paired peaks. Now it uses cross correlation. So it's going to predict fragment length, shift reads to the center of that predicted fragment length, and then effectively it's going to scan across the genome, and it's going to find regions in windows across the genome where we have an enrichment in signal in our IP versus our control. So for attack, we were scanning across with a window, and we were saying, do we have enrichment in this window versus the surrounding window? So is this region more enriched for reads per base pair than the 10,000 base pairs around it? Now we have an input control. We want to know, is my window enriched versus the same window in the IP, and perhaps also a larger window in the IP too. So as you said, there's no package for R. We can use Herper, and I think we used this in our last sessions. If anyone's having problems installing Herper, please reach out to me. I think we had some issue with some changes we made for the course, and it might have killed one of the installations. Let me know. It's certainly available to install from like our GitHub, and if you're having issues there, we can solve that too. So we can use Herper. So we just need, if it is available on Bioconductor Manager, we can do Bioconductor Manager install Herper. If you're getting it from GitHub, it's DevTools install GitHub Herper. But once we have it, we can load the library Herper. And Herper allows us to interact with the Anaconda package management system. If we can remember what that is, Anaconda is just, it's both a tool, but also a set of libraries which allow us to install things like SAMtools or Max. And the nice thing is that you can just tell it to install a particular external software, and it creates a little self-contained environment where you can install that software and all its dependencies. So what Herper does above that is once you've installed it, well, it will handle all the installation, etc. So here we're going to do install Conda tools. I'll tell it I want Max2, and I'll tell it to install it within an environment called Chipseq analysis. And then it goes off. If you haven't got Miniconda installed, it will install it. And then it will create an environment inside where it's installed Miniconda. That environment is called Chipseq analysis, and it contains Max2, but also all the dependencies for Max2. Nice thing about Herper, as we'll see, is that we can then mount this environment temporarily. We don't need to change any files on our computer. To run Max, well, we're going to have to use it in a slightly different mode to previously. I think previously we gave it BAM-PE. Here we're just going to do Max2 call peak, tell it the name of the output and the output directory. And again, in contrast to earlier on, we're going to give it both a target file, so this is our sample IP file, and our control file to compare to. You can run max peak without max call peaks without an input sample. That's what we did for AttackSeq. For ChIP-seq, I don't recommend that at all. Try and find an input sample. So then with Herper, we can do this kind of easily. We'll just do my chip. We'll give it the ChIP-seq sample, my control, we'll give it the BAM for the control sample. And then we'll do with Conda environment. I tell it the environment I installed our tools in. So that was ChIP-seq analysis. So it's now going to mount that environment temporarily just while it runs the rest of this command. And then we're going to run a system command, system2, give it the max2 with these arguments. And those should be the same arguments as I've just listed here, but now from within R. The important thing here is the system command will call max, but by using this with conda env temporarily, just up until that bracket there, all the dependencies for max2 will be available to the R session. Right? As soon as this call is done, they all get removed from the environment. So max peak calls can then be found in the directory we specified here. So this directory here. Max, as we said before, it comes with a few outputs which are useful to us. It has a summits file. It has a narrow peak file, hopefully we previously talked about. And it has this .xls file. So the .xls file isn't an Excel file. It is a tab separated file pretending to be an XLS file. But it does contain a lot of what we would want for our downstream analysis. First thing it has is actually in the first lines, if we just do read delim, it has all the information on the peak call we've just made. So it has the actual command we ran, it has the arguments listed here, and then it has some additional information following this. What we really want to get to, though, I mean, that's great for our records to have that, but we want to get to the actual peak calls in that file. So we saw that we can use comment chars to skip lines. If we look here, we can see all these lines start with our standard comment hash. So we can do read delim, and we can tell it the comment char is a hash. And then I can read these XLS files by just doing standard base R, right? Because they're not XLS files, they're tab separated files. If we want to really deal with XLS files, we have the RIO package, which allows us to import and export XLS. So I have my data frame, and it has everything I think I would need for further analysis. Probably want to call that or change that into a G-ranges pretty quickly. So I'll load genomic ranges. I'm going to pass the chromosome column to the seq names part of the G-ranges function. I'll build my I-ranges from the start and the end columns. And then I have my G-ranges of max peak calls here. I can, we know how to interact with them, so I can get my seq names from my G-ranges, my ranges from my G-ranges very quickly. But if I want to add some more information to my G-ranges, I'm going to use the mcols argument, give it my G-ranges, and tell it what else I want to attach to that particular metadata columns. Here I'm going to do absolute summit, because absolute summit's where we're going to look for motifs. And fold enrichment, because fold enrichment gives us a very simple metric to rank our peaks by how much stronger they were than the control. Okay, so we can rank peaks by fold enrichment, and we know the center of the peak by the absolute summit position. We could also actually import the narrow peak file. So I didn't really use this much until very recently, where our track layer added a format called narrow peak. And then I can just import that directly then, the narrow peak file, rather than the XLS file. And then I have a G-ranges object, and it has most of the information we need, has our, well, all the information we need. Seq names, or the peak position, seq names, ranges, the name of the peak, the score of the peak, the signal value, the p value, the q value, and then the peak number. So just 1 to the total number of peaks. First thing we want to do, and we did this in Blacklit in the AttackSeq as well, every time you've called peaks, always make sure you remove any peaks which overlap any artifact regions you're aware of, be it the common blacklist or your gray list if to find one. So here I'm going to import our blacklist from earlier into the import bed function, and then I can just overlap my peaks with my blacklist, take those which don't overlap, and use that to subset my peak set. So this is going to give me true or false. This range overlaps the blacklist, and then it will reverse the true or false with the exclamation mark, and then I can use that to index my G ranges. So I think the final thing we're going to do then is peak annotation. So I've got my peak calls, I've assessed my ChIP-QC quality, I've assessed my FastQ quality earlier. I next want to say, well, if this is a transcription factor and it's regulating transcription, let me know which gene is potentially regulated. So that's hard to do without things like Hi-C 3D data. So I actually know that this enhancer or this transcription factor is physically connected to the promoter. Without that information, we can just do a few simple sets of rules to annotate genes to peaks. One is they overlap a gene. Right? We could assume that if a peak lands in a gene, it's probably regulating it. Or we could say the gene is closest to a peak, and that probably could say this peak is regulating this gene because it's the closest gene to it. And then we can just set some rules like a minimum distance. So if a peak is in the middle of nowhere, we won't believe it regulates a gene 200,000 base pairs away. But if it's nearest to a gene 2,000 base pairs away, 10,000 base pairs away, we might believe that's regulating. So we set a minimum distance here. So we can use the ChIP-seq package for this. There's also ChIP-peak anno, another package, but we're going to use ChIP-seq because it has lots of ways of plotting our data, and it works well with Bioconductor objects. So we're going to load some packages we need. We'll load the TXDB for mouse. We'll load the organism DB. So this contains our gene names and our entrace gene names and our gene symbols will be in there. We'll do the Chipseq library. That's actually going to do the work for us. And we'll import genome info DB, because actually this is the tool which allowed us to change chromosome kind of namings. So we can move from a UCSC style of genome names to an ensemble style of genome names, or DB gap style of names. It wasn't DB gap, it was the SNP database, DB SNP style of names. Okay, so we're going to do annotate peak. We can give it our, we should have seen this before. This is from our Chipseq library. We saw this in attack. I'm going to give it our G ranges. I'm going to give it the TXDB object, and I'm going to give it the org.mm.eg.db object. You don't need to give it this org.mm.eg.db object, but you might not have them, right, if you're using custom genomes, because you could build this object yourself. TXDB from GTF will allow you to do that, but you can't make this yourself. So if you don't provide this, you can just merge in your own gene names. But if you do provide this, it's going to not just give you entrace gene IDs, it will give you gene symbols as well. Just so much more easier to translate, I guess, understand. So anyway, we run this, and it's going to perform our annotation as you see here, and it gives us back a CS anno object. If we just type that into the screen, the CS anno object, it gives us a nice overall of what's happening, and it tells us, 41% of peaks land in promoters, 13% land in first intron. So just by typing it in, it gives us like total number of peaks and then percentages of their breakup in annotation. Worth noting here that this is impossible, right? You can't. This adds up to 100, which isn't possible, because promoters do overlap other transcripts, 5' UTRs, right? A first intron might also contain a promoter from another gene. Downstream will certainly contain other genes. So what they've done here in order to provide this 100% percentage is once a peak is associated to a promoter, it can't be associated to anything else. And the order of association is the order you see here. So once you're a promoter, you're defined as a promoter. It doesn't matter if you also overlap another gene's 5' UTR. If you're a 3' UTR and none of the above, you can't then be one of these. And that's how it's this order of annotation, which we see here, is how it can add up to 100%, because annotations overlap, they're not distinct. This is great, this overview, but actually I probably want to get down to a peak to gene level of annotation. We can do that by either turning our CSAnno object, what we created here, into a granges. So if I do as.granges on this, I can get a granges of the object, or I can do as.dataframe and get a data frame of the object. And the data frame is perhaps more useful to write out to. When I look at the granges, this will be very similar to what's in the data frame. We can see the information we have, our peak, the absolute summit's still there, the fold enrichment's still there, but then I have the parts from Chipseq, so the annotation region, the actual gene it's annotated to, the transcript ID, the distance from the peak to this gene's TSS, and then we have additional gene kind of information here. So this is what came from the org-db package we supplied, ensemble, symbol, and gene name for the entrees ID for this gene. There's the ID. Nice thing about CSAnno objects and Chipseq is that they have some kind of cool ways of plotting it. So we can do a plot anno bar, and that will kind of break up our plot we saw earlier on. So this thing we saw here, it will put it into a bar style plot for us. We can also do plot anno pi. People don't like pie charts, but if you wanted one, you can create a pie chart from this too. Another nice plot we could do is plot distance to TSS, and this kind of gives you an idea about how far away your peaks are in a more qualitative way. And it kind of breaks up your bins into 0 to 1K, 1K to 3K, and then getting larger bins. It just shows you the proportion of data which is in that. Okay. So if you added all of this together, you get 100, but it's trying to centering it around the TSS. So on this side, we have less than 50, it's like 45, but on this side, we have 50 something. Another really useful plot. So I talked about overlapping annotation. So you can actually get an idea of how overlapping the annotation is from using this upset plot, peak anno, give it our CS thing. Here I'm just saying then pi equals false. But here along this axis, we can see genic and intron overlap quite a lot. And we have intergenic and distal intergenic clearly overlap as well. I mean, that makes sense. But then we have genic, intron, promoter, exon, and 5-UTR also overlap a lot. And there are this many which overlap from that term. So we can get an idea of the overlaps in our data, how much, which terms or which sets particularly overlap, just using this upset plot with our peak anno. That's the end of our session 2, I think just about in time. We have some exercises here, and this is, so I've been showing you MIC. This is Chipseq quality from CTCF, just to give you an idea of the, you can't even see the phantom peak here. It's so small compared to the actual signal. So it's worth having a little go at that. A little bit of ChIP-QC plots, and then some annotation. Some other plots coming. So if you want to have a go at that, one more round if you want to ask me any questions, and I will try and upload all the videos. Hopefully today I'll upload the attack, and I'll try and get the other data on as soon as I can. The other videos on as soon as I can. So that's the end of the session for today.